Hello, my name is Kevin Halling. I'm a molecular pathologist at the Mayo Clinic. I'm going to talk to you today about the use of RNA-seq for detecting gene fusions in tumors. A little background about myself. I did my pathology residency at the Mayo Clinic from 1991 to 1995. I then started a molecular diagnostics lab down at Indiana University for a few years. And then I came back to Mayo and came onto staff in 2000. And I've been part of the Genomics Laboratory ever since 2000. My main interest has been in developing and implementing assays that are used for to help manage patients with cancer. So that's why I got into this particular area. So today we'll be focusing in on using RNA-seq to detect gene fusions in tumors. I have no relevant disclosures for this talk. And the overview of my presentation is this. So we'll start a little bit about talking about basic cancer concepts, and then we'll talk about the clinical importance of gene fusion detection. Then I'll follow that with a discussion of the methodology that's used, basically the chemistries that are used in the bioinformatic analysis. And then I'll finish with a clinical case that sort of illustrates how we clinically interpret gene fusions that are detected by RNA-seq. So I think everybody knows this, but just to remind ourselves, cancer is a genetic disease, and it results from mutations that, in two types of genes, oncogenes and tumor suppressor genes. So as you probably also all know, there's about 20,000 protein encoding genes in the human genome, but only a small fraction of these, about 500 to 1,000, have the ability to contribute to cancer formation when mutated. So some genes, for instance, when they're mutated, such as hemoglobin, collagen, albumin, and keratin, are unlikely to contribute to tumorigenesis. The mutations in oncogenes like KRAS and EGFR and tumor suppressor genes like P10 and P53 do contribute to tumor formation when they're mutated. So again, to reiterate, there's two types of genes that cause cancer, oncogenes and tumor suppressor genes, and they do this in sort of opposite fashions. So oncogenes contribute to tumor formation through activating mutations. In other words, mutations that increase the activity of the proteins that they encode. And tumor suppressor genes contribute to tumor formation through inactivating mutations. In other words, mutations that turn off the function of the proteins that they encode. There's different forms of mutations. Some occur at the chromosomal level, and some occur at the molecular genetic level. So examples of chromosome level mutations include translocations, interstitial deletions, inversions, and amplifications. So an example of a chromosomal alteration is shown off to the right. That's the famous Philadelphia chromosome. Examples of molecular genetic level mutations are point mutations and small insertions and deletions. So as I said, I'm a molecular pathologist, and my job is basically to develop tests and implement tests that help us identify these mutations to help manage the care of patients. So the type of test we use depends on whether it's a chromosomal level mutation or molecular level mutation. And some examples of tests that are used to detect chromosomal level mutations include karyotyping, chromosomal microarray analysis, fluorescence in situ hybridization, pair next-gen sequencing, and, as I will talk about today, RNA-seq for gene fusion detection. Example molecular level mutation assays include PCR-based assays, Sanger sequencing, DNA next-gen sequencing, and you can also use RNA-seq to pick up molecular level mutations, but we won't be talking about that today. So this slide just shows examples of some of the data we get from the different assays that we use. So an example of microarray analysis shown at the upper left, of Sanger sequencing at the upper right, of next-gen sequencing at the lower right, and FISH at the lower left. So why do we care about picking up these mutations? Well, as I said, it can be very clinically useful. It can help establish a definitive diagnosis for a patient with cancer. It can help determine what their prognosis is going to be, and it can help guide therapy, especially for targeted therapies. So as I said, we'll focus today on the use of RNA-seq for gene fusion detection in tumors. So let's start with the definition of what a gene fusion is. A gene fusion is a fusion of two genes that results from a chromosomal rearrangement, such as a translocation or an interstitial deletion or an inversion or an amplification. And one of the best known examples, and the very first example, was the gene fusion that results from the Philadelphia chromosome that occurs in patients with chronic myelogenous leukemia. This was the very first genetic alteration that was ever discovered in a tumor way back in the 60s by Janet Rowley. And she found that there's a translocation in over 95% of CML cases that joins the long arm of chromosome 22 to the long arm of chromosome 9. And it wasn't known at that time, but a decade or two later, they discovered that what was happening at the molecular level was a gene fusion, which joined BCR, the BCR gene with the able oncogene. So gene fusions are often a mechanism by which oncogenes are turned on or activated and promote tumorigenesis. Gene fusions are common in tumors. They're more common in some tumor types than others. They're quite common in sarcomas. Maybe 30 to 40% of sarcomas are found to have a gene fusion. A high proportion of hematologic malignancies, but only about 5% of lung cancers, for instance, have a gene fusion. And as I mentioned, gene fusions can be helpful for establishing a diagnosis, guiding or determining prognosis and guiding therapy. Certain gene fusions are specific to certain types of sarcomas, hematologic malignancies, and salivary gland tumors. And because they're specific, they can be used to help establish a diagnosis. So a good example is a type of sarcoma known as Ewing sarcoma frequently has fusions that involve the EWSR1 gene with other gene partners. So EWSR1, FLI1, EWSR1 ERG are characteristic of Ewing sarcoma. And so if a pathologist is wondering if the tumor he's dealing with is a Ewing sarcoma, he may order an assay that will help identify these fusions. Another good example is a PAX3 FKHR fusion that's seen in alveolar rhabdomyosarcoma. I mentioned the BCR-ABL fusion in chronic myelogenous leukemia. A couple that are useful for diagnosing salivary gland tumors are shown at the bottom there. CRTC1 and MAML2 is seen in mucoepidermoid carcinoma, and MIB-NFIB is seen in adenoid cystic carcinoma. Now, the next slide here shows a not uncommon scenario that a pathologist faces. They may be looking down the microscope and seeing something like you see off to the right there, which they would describe as a small, round, blue cell tumor. The problem they have is that this could be any one of the number of different types of tumors that are listed off to the left. It could be a Ewing sarcoma, it could be a peripheral neuroectodermal tumor, it could be a rhabdomyosarcoma, and the list goes on. So they may take into account certain, the patient's clinical features, for instance, the age, the gender, where the tumor is located, and such. And they may also apply a battery of immunostains to help make a diagnosis. But sometimes, despite all of that, they're still left with not really knowing what it is. And they may feel that what would really help is having an assay that would look for a gene fusion that's characteristic for a particular type of tumor. How do we traditionally detect gene fusions? Well, in the past, what we've done is one of two assays, either RT-PCR, which stands for reverse transcriptase PCR, shown at the top, or FISH, fluorescence in situ hybridization. With RT-PCR, what you do basically is you isolate RNA from the tumor, you convert it to DNA through reverse transcription, so that's the RT part, and then you do a PCR reaction with primers that are designed to pick up the fusions you're looking for. So in this case, what they were looking for was a JAS-F1 SUS12 fusion, and so they did their PCR reaction with these primers, and they found a fusion which was, they could demonstrate on a agarose gel here, and then they sequenced it and it fits. So this is how it works with RT-PCR. The difficulty with RT-PCR is you have to know the two different partners generally to make it work. FISH has the advantage of not having to know both partners. All you have to know is one partner. And so at the bottom, we show basically what FISH does is it uses a break-apart FISH program strategy, where you use a 5' probe that's colored green and a 3' probe that's colored red. And if there's a rearrangement, such as occurs with a fusion, these two probes will break apart, and you'll have separate red and green probes. If there's no break apart due to a rearrangement, they'll be right on top of each other and appear as a yellow signal. So FISH, again, has the advantage of not having to know what the fusion partner is. So that's both an advantage and a disadvantage, because if you want to know what the partner is, you won't be able to tell. Only RT-PCR will be able to tell you that. So this is traditionally how we picked up gene fusions. It's limited to being able to pick up just 1 or 2 gene fusions at a time. The advantage of RNA-seq is that you can simultaneously pick up fusions in the 20,000 genes if you wanted, any one of 20,000 genes simultaneously. So it's much more powerful. This slide sort of further illustrates the shortcomings of RT-PCR for detecting gene fusions. So we know in Ewing sarcoma that the two most common fusions are EWSR1 Fly1, which accounts for about 85% of tumors, and EWSR1 and ERG, which accounts for about 10% of tumors. But then there's a variety of other fusions that account for just a few percent of tumors. It's impossible to easily make primer pairs for all of these, so we just designed an assay for the top 2. And so you can see that the problem here is that you're going to have an inherent false negative rate of at least 5%. So we knew with our RT-PCR assays that there's a potential to miss rare fusions. And RNA-seq, again, doesn't have that limitation. It could pick up all of these fusions. The other clinical utility of RNA-seq for fusion detection is detecting gene fusions that can help guide targeted therapy decisions. The best example here is for lung cancer. About 5% of lung adenocarcinomas have a fusion involving either the ALK oncogene, the ROS oncogene, or the red oncogene. And the value of picking up these gene fusions is that, for instance, if you had an ALK or a ROS gene fusion, that would predict the patient would respond to a drug known as crizotinib or similar drugs. If the tumor had a RET gene fusion, it would predict that the patient would respond to a drug known as vandetanib. But the problems, for instance, for picking up ALK fusions in lung cancer are the same as I sort of described for EWSR1 fusions in sarcomas. So we know with ALK fusions in lung cancer that there's a variety of 5' partners. For instance, you can have an EML ALK fusion, a KIF5B ALK fusion, NBAS ALK fusion, so a variety of 5' partners. And so, again, making it difficult to design primers for all the different possible partners. In addition, the breakpoints can vary. So usually with ALK, it's an exon 20, but with the 5' partners, it can be all over the place. So all of this illustrates the difficulties of designing RT-PCR assays to pick up all these different possible fusions. And FISH suffers from the limitation of you would be able to pick up most ALK rearrangements, but it wouldn't tell you what the partner was. And also, sometimes there's funny FISH patterns that result that are not really due to fusions. All right, so we've been interested in this since about 2015. We started working on RNA-seq for gene fusion detection back then. We decided we wanted to use whole messenger RNA transcriptome sequencing. In other words, doing RNA-seq on the entire messenger RNA complement. But as I said, that's about 20,000 genes. We didn't want to look through all that data. We just wanted to look at the genes we were interested in, and that was 571 genes. So we get data for 20,000 genes, but we bioinformatically mine out just the data for 571 genes of interest. We went live with this assay in January 2018. We're currently only offering this internally to Mayo patients because it uses snap frozen tumor tissue or tumor cultures, and logistically it's difficult to send those in. We hope next year to be able to offer RNA-seq on FFPE specimens, and that would allow us to open it up to outside laboratories and hospitals. As I mentioned, we do RNA-seq on 20,000 genes, but we only are interested in the results from 571 genes. And the beginning of that list of 571 genes is shown here. The way we came up with this 571 genes is we went through the literature to see what gene fusions have been described in different tumor types, and then even beyond that, what known oncogenes exist. So it's pretty comprehensive. So how do we perform RNA-seq? Well, the first thing you need to do is obtain a tumor specimen. You then isolate total RNA. And total RNA, as you remember, consists of ribosomal RNA, messenger RNA, transfer RNA, microRNA, and other types of RNA. But we're just interested for our test in the messenger RNA. So we have to find a way to specifically isolate the messenger RNA out from the total RNA. And the way we do that is we take advantage of the fact that messenger RNAs have a poly A tail. So we use oligo-DT beads to isolate the messenger RNA. Once we have the messenger RNA, we then convert it to cDNA through reverse transcription. And at that point, it basically becomes very similar to DNA next-gen sequencing. So we prepare our libraries. We put those libraries on a next-gen sequencing instrument, and we use Illumina instruments. And then we get a lot of data. And that data would be impossible to go through manually to look for fusions. So we have a bioinformatic pipeline that crunches that data and basically sort of spits out the possible fusions that exist. And then finally, we have to go through that list, maybe a small list or a bigger list, to decide which things are important and need to be reported. So next-gen sequencing generates tens to hundreds of millions of reads. And what we do is we align, depending on the type of what we're trying to accomplish, we align those reads either to the human genome, the transcriptome, or just a specific set of genes. And we have to use customized bioinformatic pipelines to determine if there are mutations present in the reads. Different types of pipelines are required for different types of mutations. So typically for DNA next-gen sequencing, where you're looking for a point mutation or indels, you'd use one type of pipeline. We use an entirely different type of pipeline for gene fusion detection. And another type of pipeline might be required if you're looking for copy number alterations. This shows an example of DNA next-gen sequencing, just to illustrate sort of how next-gen sequencing bioinformatic analysis works. So as I said, you get a lot of reads, and this is a lung cancer sample that was sequenced from a patient. And the sequencing reads are shown below these sort of blue lines. Each line represents a single read, so there's lots of reads, and they keep on going below the screen. And the reads are aligned to a gene of interest. So in this case, we're interested in seeing if there's any mutations in the EGFR gene. And if there's no changes shown below, there's no mutation. But if there's a change shown, and you can see, if you look carefully, you'll see a G in some of these reads, that means there's an alteration. So at this position, there's a T to G change in about 20% of the reads. And that changes an amino acid in EGFR from leucine to arginine, codon 858. And that's a well-known mutation that predicts that the patient will respond to EGFR therapies, anti-EGFR therapies. So that's sort of how it works with DNA next-gen sequencing. The bioinformatic pipeline for RNA-seq is quite different. We're actually looking for reads that don't map to the transcriptome. You wouldn't expect them to map to the transcriptome because they're not normal. They have a fusion of 2 different genes. So we take the reads that are initially thrown out and then look at those to see if there are reads that span 2 different genes. And when we see these reads, we call them supporting reads. They can take one of several forms. So they have different definitions. Remember, this is paired in next-gen sequencing. So we get 2 reads for every read. We get one in this direction, one in that direction. If both of them span a junction of 2 genes, we call them spanning reads. If they're entirely contained in the separate genes, we call those split reads. And if one crosses a junction, the other is entirely in the other gene, we call that a hybrid read. It's not all that important that you understand the difference between these, but just realize if we see 5 or more of these collectively, we can call it the case positive for a gene fusion. So we call, again, these are supporting reads. If we see 5 or more supporting reads, we consider the case positive for a gene fusion. And this is an example of a real life case that where we picked up a novel fusion in a thyroid cancer. It was a fusion between a gene known as STXB6 and the oncogene BRAF. This fusion had never been described, but other BRAF fusions have been described in thyroid cancer. And down below, you can see these are the individual reads that are spanning the junction between the 2 genes. We always confirm our fusions with RT-PCR just to make sure they're real. And I'll tell you that probably over 98% of the time they confirm. So the assay has very high specificity, not a lot of false positive results. And this just shows an example of a fusion we picked up in a liposarcoma between a gene known as MDM2 and TMPO, and it joined exon 7 of MDM2 to exon 4 of TMPO. And we confirmed it by agarose gel electrophoresis and also sequencing the products. Now, the bioinformatics is really crucial to this because we would not be able to handle all of the data we get without a bioinformatic pipeline. And what our bioinformatic pipeline does in part is to filter out all the junk. So this is an example of a case that was a hematologic tumor, was an acute myelogenous leukemia, that we picked up a fusion between a gene known as RUNX1 and RUNX1T1. But if you look at the bioinformatic analysis, on first pass, it looked like there was a potential for 129 fusions, way more than we can handle interpreting. But if you then ask how many of those 129 fusions overlapped with our 571 genes of interest, it was only 10 genes. So we got all the way from 129 down to 10. If we then ask how many of those 10 fusions had 5 or more supporting reads, we got it down to 5. And we have other things we consider, for instance, is it ever seen in normal tissues? And is it an exon to exon in-frame fusion? So ultimately, we got it all the way down from 129 possible fusions down to just 2, which is very manageable. And actually, the 2 fusions were the RUNX1, RUNX1T1, and the reciprocal translocation. So the bioinformatics just works wonderfully to get it down to a manageable number. This slide sort of illustrates that. So 58 tumors that we analyzed, looking for fusions. We found in about a dozen cases no fusion. In about 33 cases, we found 1 fusion. About a dozen cases, we found 2 fusions. And just 1 case had 3 fusions. So the point of this slide is just to show you that we detected anywhere from 0 to 3 fusions, which is quite manageable in terms of interpretation. Of course, as a clinical laboratorian, we have to make sure that our tests are accurate and reproducible. So we do what we refer to as verification, which is basically to establish the performance characteristics of the assay. We determine the accuracy of the assay by determining the sensitivity and the specificity. In this case, what we did, we looked at 41 tumors that we knew had a fusion based on other reference methods such as FISH or RT-PCR or RNA-seq in another lab. And with our method, we were able to pick up 38 of the 41 fusions for a sensitivity of 93%, so 3 false negatives. We also looked at 15 normal tissue specimens and 10 tumor specimens that we knew should be negative for fusions, and we didn't pick up any fusions in those samples for 100% specificity. We also did reproducibility studies, both inter-assay and intra-assay, and we found that we had 100% reproducibility. We also did limited detection studies where we made dilutions of tumor to see how low we could go. And we generally found we could go down to at least 12.5% tumor, and sometimes as little as 3% tumor, depending on how heavily the fusion was expressed. Okay, so I mentioned that we had 3 cases, 3 false negative cases up here with our sensitivity, but it's important to realize that we also picked up 22 fusions in 17 tumor specimens that had not been detected by other methods. So there was a big net gain in total number of fusions detected. And 18 of those 22 fusions were novel fusions that had not been previously observed or described in the literature. And we confirmed all of them by RT-PCR. Now, not all of those fusions, 22, are necessarily clinically significant, and I'll talk a little bit about that in a second. They're real, biologically real, but whether they're clinically significant is a different question. So we described our verification studies in a paper in Journal of Molecular Diagnostics in 2018, if you're interested in reading more. Okay, the final thing I want to touch on is how do we interpret gene fusions that are detected by RNA-seq? In other words, how do we know that they're clinically important? As I said, not all gene fusions detected by RNA-seq are clinically significant. Occasionally, some of them may represent technical artifacts, and we hope that the bioinformatics weeds out most of those. And some may be real fusions, but clinically insignificant, what we refer to as passenger mutations that result from genomic instability in cancers. And so the next few slides will use a case study to illustrate how we determine if a fusion is clinically significant using a combination of bioinformatics and clinical interpretation that's based on databases and literature. So this is a very interesting case. This was actually the first case we ran through the clinical assay. It was a 72-year-old man who had a destructive 4-centimeter tumor of the mandible. The pathologist diagnosed it as a spindle cell sclerosing rhabdomyosarcoma. And this was the fusion filtering process for that particular case. The potential for 194 fusions, but whittled all the way down to 1 real fusion. And I'm just showing you that. So 194 possible fusions, only 8 of them overlapped with the 571 genes of interest. Only 1 had 5 or more supporting reads. There were none of them that one didn't have was not a read-through event, which means occasionally you see transcription reading through genes goes from one gene to the next, and those can appear as artifacts. So that wasn't like that for this case. We didn't observe this particular fusion in any normal specimens, so it's not a benign thing. And finally, it was in frame. So bottom line is 194 fusions whittled down to 1 real fusion. And this is what it looked like. So it was a fusion between an oncogene known as FUS and a transcription factor known as TFCP2. It joined exon 1 through 6 of FUS to exons 2 through 15 of TFCP2. This fusion had never been described before. FUS fusions had been described, but never with this gene. We confirmed the fusion by RT-PCR, shown off to the left as a band on the agarose gel. And so the next thing I did was I went to the databases to see if this fusion had ever been described before. And while FUS fusions have been described, this particular FUS fusion has never been described. So you can see some of the FUS fusions that have been described, such as FUS CREB301 and 2, FUS DDIT3, FUS ERG. And this is just showing, these columns are showing you how common they are. So the most common FUS fusions are FUS DDIT3 and CREB301 and 2. And these FUS fusions are observed in different types of tumors. For instance, FUS DDIT3 is observed in myxoid liposarcoma, and actually finding this fusion is very helpful for establishing a diagnosis. But as I said, the fusion we saw was never described before in the literature until, ironically, just a few days later, when we saw a paper come in, into the literature out of Europe that described a completely novel group of epithelial or spindle cell rhabdomyosarcomas that were characterized by FUS and TFCP2 fusions. So brand new. So this is when you see sarcomas with this particular type of fusion, it's a particular type of rhabdomyosarcoma. And we described our experience in a paper a few months later. So that's all of my slides. So just to summarize, RNA-seq is a powerful tool for detecting gene fusions in tumors. And clinically, it can be used to help establish a diagnosis, especially for sarcomas and hematologic tumors. And it can help guide therapy. Basically, it can help determine if patients are candidates for targeted therapy. Best example being ALK inhibitors in lung cancer. But there's many other examples. And then finally, I hope I showed you the process that we use to determine if gene fusions are clinically significant, because some can represent technical artifacts that we try to weed out with bioinformatics. And others, while they may be real, biologically real, may not be clinically significant because they may represent passenger alterations that result from genomic instability. And the distinguishing of clinically significant from insignificant is highly dependent on our bioinformatics and our post-analytical clinical interpretation. So I hope I've shown you that RNA-seq is a very powerful tool, and I think that it's going to be used quite commonly for patients with advanced cancers in the coming decade. Thank you very much.